Today I am going to talk about Ziehl Neelsen stain or acid-fast stain. After watching this video, you will know about the purpose of the stain, theory behind the stain, principle, requirements, observations, as well as the special considerations when you need to follow when stain with this staining procedure. This demonstrates the characteristics of acid fastness in certain bacteria and the cyst of Cryptosporidium and Isospora. Most important application is to detect Mycobacterium tuberculosis in sputum samples. This staining method is a screening test for the rule out of diagnosis of tuberculosis in patients. There are three common acid-fast staining methods. They are Ziehl Neelsen hot method, Kenyon cold method, Orman-Rhodomayan chlorochrome method or Truant method. The slides stained by Ziehl Neelsen staining method can be visualized by standard bright field microscope or light microscope. Many bacterial cells can be easily stained with simple stains such as gram stain. However, some bacteria such as Mycobacteria and some Nocardia species do not stain using these simple staining methods. If stained, they produce variable reactions as their cell walls are not permeable to the rosanilline dyes in common staining regimens. That means the cell walls of Mycobacteria and Nocardia species do not permeable to the normal dyes. The cell walls of Mycobacteria contain mycolic acid, giving the cell wall high lipid content. That means in the cell wall of Mycobacteria contain mycolic acid. This characteristic is thought to be the reason these bacteria are difficult to stain with simple dyes. To stain these bacteria, stain require high concentrations of dye solutions, or there should be a heating period. However, once a stain is introduced into the cell wall, decolorizing is also more difficult in these organisms. This is the principle behind the staining procedure. The high amounts of lipids, I told you there are high amount of lipids in the cell walls of these Mycobacteria-like organisms. The lipid contained in the cell wall is beta-hydroxycarboxylic acid. The cell wall of the acid-fast organisms take up carbylfructine, which is basic. The cell wall of the acid-fast bacilli take the carbylfructine into their cell wall. Carbylfructine is a basic dye. Heating period enhance the uptake of staining solution and enhance the staining procedure by making strong complex which cannot be easily decolorized. This strong complex cannot be easily decolorized with diluted acids. Acid-fast organisms take up the red color of the primary dye, carbylfructine, while non-acid-fast organisms easily decolorize with acid alcohol. Methylene blue is the counter stain which stain the background in blue color. Malachite green also can be used as a counter stain. The requirements of this staining procedure: we need strong carbylfructine, acid alcohol, blue plus methylene blue. Carbylfructine, acid alcohol, methylene blue. Now we will see the method. Fix the air-dried smears by passing slowly through the flame 3 times. I think you know how to prepare the smears, and after preparing you want to allow them air dry and then fix by passing the slides through a flame. Then cover the slide with strong carbofuction and heat until steam rises. But you want to remember, do not boil it. Allow to stand for 5 minutes. Apply heat at intervals to keep the stain steamy. Do not allow stain to dry on the slide. Add more carbofuction if required. Wash with running tap water. Decolorize in acid alcohol until the traces of red have disappeared from the film. Wash with water. Then continue the above process until film gets faintly pink. Wash well in water when decolorization is completed. Counter stain with methylene blue or 0.5% malachite green for 1 minute. Then wash and air dry. When you observe the stain slides under microscope, you can see like this. This is a picture under light microscope. Acid-fast organisms stain in red or pink color. You can see here acid-fast bacilli in this picture. The other organisms may stain in blue or green color. It depends on the background stain you use, the counter stain you used. Now, this is a picture which has stain with methylene blue. Depending on the counter stain, the background get blue color when stained with methylene blue. This background get green color when you stain with malachite green. There are special considerations you want to follow when stain with this stain. When you stain in, do not use staining jars for the purpose, because tubercular bacilli from positive samples can be detached and float in the staining jars. Then when you add slides into the staining jars, the floating detached particles can go and attach to the slides. This could cause false positive results. Thank you very much.